This sounds to me like it's on. You know, I'm giving a keynote speech today, but I've been in a room with people giving brilliant talks, and I assume that what happened is that Philip drew at random from these incredible speakers that I've heard today, and it's a great honor to be able to go ahead and give the final talk of the day and tell you a little bit about my perceptions about what's going on in what is becoming living architectures. When I first met Philip, the title living architecture to me as a life scientist is a very provocative term. And we've heard in the lectures at the Bridge today, and just this evening from a few life scientists, but Philip and I entered a discussion about what it really meant to have a living architecture and what was a feature of it that might make it alive. And this is, without being pejorative, what I see in these works has been biomimicry. And biomimicry is very important, biomimicry is very instructive, but living things follow their own rules. And what I'm going to do today is spend a little time talking to you about living things and some features of living things. And it's intended mostly for the purpose of being provocative to how to think about where these things might go in the future. I'm going to talk about a few things about biology, some of the biological processes that living things undergo, the organization of living systems, and yes, I'll recap the discussion about worms. I'm going to say a few words about community and communication, but there was so much to talk about, I didn't manage to get a lot of slides on this, so it's a good slight ad lib. And then I want to talk briefly about consciousness and the internal life of organisms. It's fun, actually, that this was my first slide even before I heard Philip open up the lectures talking about the second law of thermodynamics, which briefly stated means that entropy always increases. That is, that a state where everything is in a known place is so unlikely as to be out of balance, and entropy causes things to move into more likely states. The classic visualization, let me go back here. So entropy is defined as a thermodynamic quantity representing the unavailability of a system's thermal energy for conversion into mechanical work, often interpreted as the degree of disorder or randomness in the system. We're going to talk towards the end about likelihood and energy, but this is from the energetic position, and essentially it's a system which is disordered. And so one visualization might be that I have, say, a few molecules of perfume in a space. And under random thermal interactions, what happens is these will disperse to a more likely state, which is a state of high disorder. And this is sort of typically the way we think about the second law of thermodynamics. Now, living systems are a little bizarre because they're in a constant state of disequilibrium. That is, living systems are fighting entropy at all times, and yet they're able to remain organized not just for our lifetime, but for billions of years. Single life is instantiated in you that began billions of years ago. And I kind of think about this as though life is sort of an eddy current in a wave of time and space and energy. And it's just carried forward through some bizarre process that's self-feeding on it. We know that life depends on the input of energy at all times, but why it should be able to be self-sustaining in this way is it was one of the deeper mysteries of the world. Living things fight disorder at all times. And one of the profound things about all living things is they have both an internal and an external environment, and their internal environment tends to be very tightly controlled, and it requires a terrific amount of energy to do so. So in the classic cellular organisms, which I'll really concentrate on rather than say viruses, which are maybe alive, maybe not alive, they are surrounded by a cell wall, and a cell membrane or a cell wall, and some process in the cell constantly keeps ions moving one way or the other on it, so there's a disequilibrium in the cells. One of the interesting things about this is that all cellular organisms have the feature that because they're moving ions, charged particles around, that they actually act like little batteries. And they have typically a voltage across them on the order of nearly 100 millivolts. So they're little batteries, and the batteries are being recharged by some complicated process of metabolism. In fact, the internal milieu of bodies is kept in extremely tight control, not only just within single cells, but the cells, when they're in an arrangement such as an organ, will tend to keep that whole organ's internal milieu at extremely tight control levels. So plus or minus 10%, for example, for the major ions in the environment. They do this by having a membrane around the cell which is active itself. It actually physically transports ions across it under extraordinary adversity, because in fact the electrical field across the cell membrane is a whopping 10 million volts per meter, in which you're trying to move a single charged particle. So you can imagine what sort of stresses are involved in the structure in order to make this happen. And these transmembrane carriers are themselves protein. Well, one of the things about proteins is that they're in fact relatively unstable with temperature. And so living systems actually have to maintain their temperature as well to keep the proteins from denaturing. By denaturing means they unfold and they lose their functionality. So one of the features of living systems is a need for homeostasis, a need for constant self-control and stabilization. And of course, this is an active process. Right? In the face of orders of magnitude changes in light, in temperature, and so on, our bodies are able to keep themselves under tight regulation. The cell itself is an extraordinarily complicated structure. It is so filled with proteinaceous materials and other structures in there that in fact the water component of cells is not really fluid. The water component is all tightly adhered through electric interactions, so-called van der Waals interactions, that give the water itself a stiffness which is different than water in the external world. A huge number of sort of organelles inside of cells that allow them to do this kind of complicated process. Now, one of the features of life is that living things reproduce. And we could define life all kinds of different ways, but I'm going to speak principally to life as we know it. That is, the things which we interact with which are alive. You could imagine something made out of silicon on Mars that would look alive to us maybe, but I'm talking about living things on our Earth right now. Cells reproduce. And when they reproduce, what happens is you'll take a single cell, which then divides. This is the process so-called mitosis, separating its encoding structures, its DNA, into two separate parts which are replicants of one another, and then separating into multiple cells and differentiating and growing. But one of the features of this, of course, is that that new cell is not smaller than the original cell. In fact, it is the same size in general. And so there's always a process of development within a living organism. It never is just stationary. When we talk about multicelled organisms, the process of development is rather more complicated because the cells start off in one form, which we're going to call a totipotent cell, a single cell that contains everything it needs to make an entire copy of the organism. This happens to be frog, but it may as well be human. These cells divide progressively into more and more cells during development, and then start to differentiate so the cells themselves become different types. So one of the features of multicelled organisms is the individual elements within them are highly differentiated. If you ask me a question about why something is the way it is, and people ask me these all the time, my answer 100% of the time is a simple word: evolution. Because evolution is the driving process of all living organisms. And evolution is a very curious process. And one of the things I'd like to amplify as I talk in the next couple of slides is that evolution contains its own history. That is, what we are now could not have been there had our predecessors not had a certain shape and a certain configuration. One of the ways we can observe this profoundly is under the microscope. This is Ernst Haeckel, who described the evolution of larvae of different animals. And what you can see is that the early larval stages for a human, a monkey, I don't know what a rind is. What is a rind? It's a cow? Pig? A pig is Schwein. A hen and I don't know what this guy is, a lizard, that in the early phases of development, all these organisms look effectively the same, right? And one of the ways in which we grow is that we grow out of our ancestors, and we can see our ancestors in even our own early development with this. And he coined this very interesting phrase that ontogeny recapitulates phylogeny, that in fact our growth and development really embodies within it the history of the growth and development of our entire organism. Darwin very famously saw this by looking at the differentiation of birds, in this particular case in the Galapagos Island. Each of these birds had a different feeding strategy, but they apparently had a common ancestor, a relatively recent common ancestor, and that was very obvious by the morphology of some of the features of the bird, like its beak. And all of these came from presumably some sort of he assumed it was a finch from South America, which then differentiated as these animals adapted to their environment. So these living organisms are the product of the history that created them. Now, I should just back up and once again mention, why am I talking about all this stuff? Because I want for us, when we're thinking about living architectures, to be thinking about rules of life. Evolution is one of the principal rules of life. Evolution is very interesting in many ways in that our predecessor organisms had a body plan similar to ours, but they utilized the elements of their body plan for very different things. So these are the upper appendage, what in humans we might call a hand, but in a bird, in a bat we might call a wing, in a bird we might call a wing, and these are the same bony features which are taking over different structures in the animals depending on their history. And so, you might ask why a bat has 5 apparent fingers in its wing. They don't use these things individually, but of course they've inherited that structure from some predecessor animal that walked and used those finger-like appendages. So I mentioned this notion about cells differentiating, that the original motif is pluripotent, but then evolution and our development reuses those motifs, differentiating them and finding different ways for the cells to develop and behave. And so we can think about perhaps in building structures. That's one of the things I actually love about the work that I've seen from Philip is these reusable elements, which can be built in different size and shapes, and it's very biological-like. You have a single pattern which you reuse and reutilize for different purpose. I find that very, very attractive. I'm going to spend a little time on the nervous system, so I want to show you a couple features from that. From the original sort of egg divided into 32 cells, in the process of development, the cells differentiate, and a small number of them turn out to be part of our nervous system. But there are early cells in that process, which we call stem cells, which are no longer pluripotent. They can't suddenly become a kidney cell, but they can become any neuron that they want. So again, it's like our phylogenetic tree, that there's a history that's embodied even in the individual cells in how they grow and develop. And the process of growing and developing for us, for chordates, for basically all, for chordates we'll talk about for the moment, animals with a spinal cord, is that in the early phases in those cells, we saw the cells sort of accumulate on the margin. I showed into a slide back, and I want to go and do a light show by going backwards for this. Those cells start to wrap around themselves and invaginate and create what's called the neural tube. And the neural tube is asymmetric. It has one end that's distinctly different than the other. And over the course of development, that neural tube starts to differentiate into different parts of animal brains. Now, in vertebrate brains, we end up with a spinal cord, a brainstem or hindbrain, a midbrain, and a forebrain, which is what we think about as our human brain on here. And so you get this asymmetric differentiation of the nervous system, developing into what we see as the sort of the modern, highly encephalized human brain, but it still has all the prototypical features of all other vertebrate animals. Now, the neurons themselves, these individual cells in the brain, are connected to one another in an extremely complicated manner. And we recently figured out a way to visualize that in living human organisms. This is some work from my colleagues at the Mass General Hospital, and forgetting all the equations and whatnot for this. This paper described a way of using magnetic resonance imaging, the thing we use in the clinic, in order to identify the orientation of fiber pathways in the brain. It goes roughly like this, that within the magnetic resonance scanner, it's possible to observe the diffusion of water molecules. Magnetic resonance imaging, as you may know, is based entirely on water signals. And depending on effectively the random motion of these things, you get a change in the MR signal. If you calculate the directional change, that is a diffusion along any orientation, you create a tensor, a mathematical description of the directional diffusion. And if we have a fiber mess, like might appear inside the brain, I'll show you those connections in a minute, it produces preferential locations or orientations for water molecules to diffuse. And in 3 dimensions, we talk about it as the diffusion spectrum. Because the water molecules preferentially diffuse along the directions of the fiber tracts, it's possible to infer the connectivity of individual locations in the brain by imagining the water molecules flowing along those. So here are the diffusion tensors of a large number of volume elements, voxels we call them in imaging, and the streamlines that we're showing here in color are what would happen if a water molecule entered, I can't see this thing, if a water molecule entered here, where would it most likely go? It would most likely go here. From these, it's possible to create very, very high density visualizations of the connectivity of fibers within the human brain. And so here is part of our brain called the pons. The pons happens to be one of the places where fibers from the left and right brain cross over. And you can see with these very high resolution human images where these fiber pathways go. And it's a very, very interesting structure of connectivity. But I want to talk about this from the point of view of how this works in development. So here's another visualization of this here at the basically the whole head. It's an interesting process because one of the ways that humans, or all living organisms with neurons, connect themselves is a bit counterintuitive. The neurons themselves grow these little processes which we call axons. This is driving me nuts. They grow these little processes which we call axons, which proliferate all over the place and densely connect the neurons. However, it's only over the course of life that the connectivity becomes actually relatively organized. Because what happens is that in life, that many of these high density connections become pruned and specialized. So the organism is built hyper-connected, and then through experience, the organism self-rearranges this connectivity. So the way that our brain's regions are connected becomes a developmental process based on experience. And one of the things that we see about living things is they actually change not only their behaviors as they learn, but actually their physical structure. They self-modify in the process of learning. Evolution, in many ways, is not like engineering. Evolution requires that you have a predecessor structure. Engineering doesn't. In engineering, we can make major changes in the direction of things. We can go ahead. I happen to ride a motorcycle, so I'm interested in these things. But here's a sort of version of the evolution of a motorcycle. And you might say that ontogeny recapitulates phylogeny in this case. That is, a modern motorcycle has a terrific amount in common with the first motorcycles that anybody invented, except not. Because there are these critical inventions that came from nothing that was a predecessor in the motorcycle before it, some new idea that came. And we cannot do that with living systems. Living systems just simply don't have those features. The connections between neurons. are with these little fibers, which are called axons, and I mentioned them before. And electrically, these are interesting features because what happens in these myelinated fibers is that the axon itself, the communication from one neuron to another, is covered in a very thick and dense fat layer. And that fat layer turns out to be a very, very powerful electric resistor. It impedes the flow of current across it. And so it's tempting to think about these axons as though they're some sort of a copper wire encased in some sort of a plastic insulator, and that's the way things work. And that's really the way that most people have treated neurons in here. But evolution doesn't really have that constraint. Evolution simply goes ahead and says, if it's adaptive, I'll keep it. So I've been studying electron micrographs of myelinated fibers in the brain, and I very, very often come across this motif. Let me go back. The coverings around here actually come from not the neurons, but from cells around them called glial cells. And the glial cells cover maybe a millimeter worth of axon. And then some other glial cell takes over and covers the next millimeter. And so here you can see one glial cell, another glial cell, but the axon goes through all these guys, right? At these individual crossings over here, we see a thinning, which is called a node of Ranvier. The nodes of Ranvier turn out to be interesting to electrophysiologists because that is actually the way that the electrical signals go from one end of the neuron to the other. That is, an electrical event over here does not flow through the axon, but it actually triggers a new event over here, which reforms, and then another event, and so on sequentially. Now, as I said, I've been studying the morphology of these things under the microscope, and you very often see this motif, which is that the nodes of Ranvier are aligned to one another. If you were an engineer, this is simply not the way you'd build a wiring system, right? Because what's going to happen here is there's always the possibility of current leakage between these 2 points. It's not a good insulator, it's not a good isolation system, but we're not engineered. We're not engineered. We are, in fact, developed by evolution. And somehow or other, this crosstalk between the neurons, what neurophysiologists call apoptic conduction, almost certainly is adaptive, right? One of the things that we see over and over again in evolved systems is that structures do not have well-defined functions. And I mentioned this a little earlier about when we build buildings, we could use the sewage pipes as some sort of a structural element, but we don't. That's not the way we engineer things. Evolution doesn't have that constraint. And if it turns out that I can use less bone because I have fibers connecting my muscles together that have stiffness, I'm going to have less bone. That's just the efficient way to build things. Another feature of living organisms is that we predate, we eat things. Basically all living things eat things. And built into that is a need for self-preservation and self-defense. And among the many enticing conversations I've had with Philip has been the notion of building systems which at least act as though they have a self-defensive mechanism, or maybe they actually do have a self-defense. Maybe these constructed elements try to preserve themselves by knowledge of their environment. They also respond, so I showed you in animal systems. This is now a plant system which also has this sort of self-defensive response. This is mimosa plant, and mimosa, as probably most people know, has this motor capability to it where it kind of closes up in the presence of what might be aversive or dangerous contact. And interestingly enough, it's now known that that's actually mediated electrically in very much the same way that our muscles and our neurons communicate. Living systems have the capacity for healing and self-repair. Don't bother what the slide shows a picture of. It's a very complicated process of what happens in a skin lesion. This is really just a marker for me to say that a big difference between living systems and engineered systems is that living systems contain within them the plan for their own reconstruction. And so in the face of wounds or injury, living systems heal, and they make themselves basically what they were before they were damaged. Living systems keep their homeostasis through a process, a very, very complicated process of energy exchange. And this is just a small portion of somebody's cool diagram about the metabolism of proteins and sugar molecules and the many, many chemical pathways that are involved in these living systems in order to do this. This all takes place, of course, within single cells. And it's the process by which we take the raw materials in the world, convert them into not only the structure for our bodies, but also the energetics needed in order to sustain these highly unlikely, highly dysentropic systems. Another feature of living systems is they never live in isolation. They're always in communication with their environment. And the process of symbiosis is communication of the animals or living things with other living things. And there are very, very well-known examples for this. This, for example, is an ant harvesting a farm full of algae, which it uses for nutrients, but it also, of course, supports the farming of this algae, which would not survive without the ants. This is a goby and a sightless shrimp. And it turns out that these shrimp have very poor defenses against predators, but the goby is able to fend off predators well. What the goby gets out of it is that these shrimp dig a nice little hole that becomes a cave for them. And this process of symbiosis is, of course, not just in a couple of cute examples, but it's inside our bodies at all time, that we depend on the bacteria that we feed in order to keep us alive. Another feature of living system, which I find very interesting, is symmetry. There's nothing really fundamental that I can see about why symmetry exists in living organisms. For example, it doesn't exist in all living organisms. This is a paramecium, a single-celled organism that is famously asymmetric. It looks like either your left shoe or your right shoe, depending on which side of it you're on. It's a decidedly asymmetric organism. However, evolution has tended to favor symmetric things. This is a structure so small that it's not clear that you want to call it alive. This is a viral capsid. This is the shell around a single virus. And it is an astonishingly symmetrical thing, a crystalline kind of symmetry. It's a radial kind of symmetry. But interestingly, embedded within it, principally, is DNA, is twisted pairs, which are the codes for the organization of living things, which is a wildly asymmetric structure. That is, it only twists one way. It's only a one-handed twist. And there's a deep thing about living systems that it turns out that carbon forming bonds ends up having a chirality, a particular orientation to all the bonds, so that one carbon bonded to 4 different atoms is different than the same carbon atom bonded to 4 different atoms that are reversed, right? There's like a left hand and a right hand version of these things, but living things only use one form, right? The anti-symmetric version of that doesn't appear in biology, and so there's some sort of funny dealing with symmetry that we have with biological life forms. However, for living things right now, for principal large organisms, there are 2 forms of symmetry which evolution seems to have adapted. One is radial symmetry, and the other is bilateral symmetry, shown here in flowers. And so this is not, this particular symmetry is not necessarily an issue of kingdom Animalia, but kingdom Animalia also includes both these forms. Famously, jellyfish have a radial symmetry to them, and they are very complex organisms, extremely complex organisms, which are so distantly related to us that you can track them through basically the fact that they use DNA, but there's very little about their structure which we have in common with them. Whereas organisms such as ourselves typically have bilateral symmetry. And so here's our friendly worms. The organisms which are bilaterally symmetric, the kingdom Animalia ones, which are bilaterally symmetric, seem to all follow a single canonical body plan. And it's the body plan which we probably inherited from worm-like organisms. This is an annelid. And That body plan features the fact that these worms are made up of repeated elements over and over again. I've got a little segment here. It's got, in this particular animal, which has legs, it has 2 legs on it, and then you go on to another segment which seems to be completely interchangeable with it. This is a feature of our predecessors, and therefore it's something that, because they're our predecessors, is going to be embodied within us. If you then take these guys apart, what you'll see is they have, I can't see it, a digestive system which goes right down the center of them through the tube. They have a vascular system, blood vessels, which goes down the center but has these radial features that come out from it. They also have what we're going to call a nerve cord, a collection of neurons in there, each of which originates in a way that goes down the length of the body but then spreads out to the various leg segments. This is our own spinal cord. And here you see the bones in your spine, but the spinal cord itself goes right through the bones in our spine, and you can see that it has this segmentation to it. Right? This is the body plan we live with. This is shown now throughout head to toe, in which you can see we have a countable number of segments. I think it's 42. I've forgotten what the number is in humans. But we're following that same original body plan. Here it is in dissection from von Hagen. I love his dissections. These are such gorgeous things. When Philip and I started talking about the information arrangement for the hylozoans, one of the things we talked about is what might make that more mimetic of living systems. Well, so we think a little bit about how segmented organisms work. We have a brain, which is some sort of which offers a great deal of high-level control, and our brain actually communicates segment by segment. We have a single cell, for example, in our head, which goes all the way down our spinal cord. It turns out it crosses over in your neck to the other side, and then reaches down, for example, as far as your hips, a single cell. And that cell gives a message from the brain, which is transmitted down not to the muscles, but to some other set of neurons within the spinal cord, which controls some sort of a local circuit over here. So essentially we have a distributed processing system whereby a message is sent from here to tell this region how to do its next trick, but it doesn't necessarily directly create that next trick or behavior. In fact, these individual segments can act very autonomously, and it's important that they do. One of the pieces of evidence for this is what we call a spinal reflex. Everybody has been to the doctor, all these adults have been to the doctor and had them tap on your knee and make your leg kick out. Well, what are they doing over there? They're testing whether or not the pathway that goes from your knee to your spinal cord is properly intact, because that pathway allows a sending input from your knee to go ahead and reach the spinal cord, which then causes some neuron in the spinal cord to send the leg out kicking. And it doesn't require the brain. Okay? This is an autonomous process, and there are bazillions of reflexes that have that level, but have that sort of characteristic. But these take place in the spinal cord. This is highly adaptive for us, it turns out, because it takes some time for conduction from neurons to reach your knee to your head. And this pathway of going through your spine is a much, much lower latency pathway. You get immediate responses for it. It's a very efficient way for organisms to work. It also distributes the process of computing so that local circuits over here, and now this is of course a trivial local circuit, or simple, I should say, nothing's trivial in biology, a simple local circuit consisting of a couple neurons to make a computation. But within the spinal cord, there are a huge number of very delicate computations that can be made without the involvement of the brain. And in fact, most of what goes on in our spinal cord is highly computed, highly coordinated, and beyond our conscious awareness. Right? These are things that go somehow autonomously. Fidelity is something also that Philip and I have talked about a little bit. There was some discussion about making these hylozoans create their own sound. And the question was, can I make out of this hylozoan a speaker which produces the full range of human audio perception with great fidelity? And in the end, I don't think that's very important. What the hylozoans should do is create their own internal sound. My vocal cords are low fidelity things. They have a very limited range of things they can do. They can't reproduce somebody else's voice accurately, for example. On the other hand, from the perception end, high fidelity is an extremely important process. And I'll talk about this a little bit from the context of the way an engineer might think about this. Linear systems are systems which obey a very simple law, that if I take a function, my linear system is some function which acts on an input x and produces an output f of x, right? If I take that input and scale it by some known amount, just make it twice as large, that the output is the same as I would have gotten to the smaller input, except scaled in the same manner. That's what linear systems is. That's all it is. For example, here is an input which goes through, in fact, the linear system. This happens to be convolution with some filter, and it produces a complex output. But it's linear in the sense that if I scale the input, I get a scaled version of the output. Okay. Well, why is that important? Because it is a property of linear systems that if the input contains signals of some frequencies, the output contains only signals of those same frequencies and nothing else, right? So if you imagine a nonlinear systems as one in which you get an input and you start to see components which are not directly in that input, because as you change the level of the input, it changes the content of the input. Right? That's a feature of linear systems. And it's a necessary feature for an organism which survives in a sensory environment, that it's able to transduce the environment with some degree of linearity. A simple example of a linear system is a spring. When we compress the spring with some amount of force, it changes the length in some linear manner. If I expand the spring, I get the same kind of feature over this. And that's a lovely linear system. Now, in fact, nothing in the world is really very linear over a long range. If I were to take that spring and stretch it further, eventually what I find is that I exert more and more force, but the thing fails to get any longer. Well, why? Because it kind of unravels until it reaches some point that it breaks. We live in an environment, let me skip that. Okay. We live in an environment where we have to receive information over orders and orders of magnitude of intensity. This is a plot on the left here of perceived loudness as a function of the intensity of sound inputs. And this is in a decibel scale, meaning that to go from 0 to 20 means that the actual energy increased by tenfold. By 40 dB, it's 100-fold, and in fact, we see a million-fold, we perceive a million-fold differences in sound intensity and treat it all as linear. And the way that our organism takes care of that is by having some sort of a negative feedback system which reduces the gain of the system so that everything remains within the linear range. In our ears, that's mediated through some structures inside the middle ear, which are bony structures supported by muscles that in fact change the mechanics of this system depending on how loud the sound is. So if the sound is very loud, you reduce the transduction efficiency of the system so that by the time it gets to the sensory organs of the ear inside the cochlea, the sound is reduced in intensity and operates in the linear range of that receiver. Similar thing happens in our eyes. As we increase light intensity to our eyes, the pupil contracts, and that's in order to reduce the amount of light that comes into our retina and avoid saturating our retina, so our retina remains linear. And of course, we see the same thing in cameras, which are built on the same plan as eyes. Organisms work in an environment where they communicate, and that supports our symbiotic activity from this. And I want to diverge a little bit, instead of going on to a long ad lib about this. I want to talk about another form of communication, which again, this slide is inspired by interactions with Philip, but also in my own work. Hans Berger, when he was 19 years old, was in the cavalry in Jena, and he was nearly crushed by an artillery cart. And he was in grave danger, but fortunately the horses stopped just in time, and he lived through the event. That evening, His sister, who was several hundred miles away, awoke in the middle of the night in total fear that Hans was in some sort of terrible danger. And she was so fearful of this, she went and sent a telegram out to him out in the field, which he received, at which point he realized that he and his sister had communicated by some form of extrasensory communication. And he then spent the rest of his life trying to understand this extrasensory communication. And he had read that it's possible to record electrical signals from the surface of the scalp on animals. And he reasoned that perhaps those electrical signals were the way by which he and his sister communicated. So he went on and built a lab secretly in his basement because guess what? Everybody would have thought he was nuts, just like we do today. But he pursued this thing in secret and showed that in fact you could record electrical signals from the human brain. These are from the human head, excuse me. These are actually of his son, who is his principal research subject. And what happens is that when Klaus, his son, closes his eyes, the signal on his brain starts to set up these very, very large, almost sinusoidal oscillations. When Klaus opens his eyes, these go away. And this is what we, of course, now call the human EEG. And it's used medically for all kinds of things. There's a huge interest now in people building brain-computer interfaces to say, well, shouldn't we be able to harness the EEG as a way of communicating with devices? I don't happen to be a big believer in that. However, I got very interested in the idea of what kind of communication would be possible with EEG. And so I set up an experiment on empathy. Now, it turns out that if I were to attach wires to your head and have them control the brightness of a light based on how much of this, say, 10 hertz sinusoid I saw in here, the so-called algorithm, that you would be able to control that light. That is, you would be able to sit there and work it out somehow or other to make that light brighter or darker. How many of you have done that experiment? I imagine that Alan has. Yeah, okay. So correct me if I'm wrong, but most people would say that they really have no idea how they do it, right? They can do it, but it's not as though thinking about a particular thing will get there. And in fact, my subjective experience is of being in a particular emotional state, so being kind of feeling a certain way. So I got interested in the idea that we could communicate feelings among people in a way that you can't using our other normal forms of communication. So here's the device. We connect EEG to two different subjects. We built our own little teeny EEG amplifier over here. We're trying to make this thing now to be 2 cubic inches. We process the EEG jointly from the two humans, and we analyze not only what frequencies are in it, but also the similarity of the signals across the two individuals. And we then go ahead and convey all of these parameters to a system which produces effectors, feedback systems to the people, which tells us effectively how similar their EEGs are. Now, the way we did it, the feedback is in the form of sound. We had 8 different channels of sound, each of which expressed the coherence of some different aspect of the EEG, and sort of colored lights that people wore as hats. It looks a little like this in the version of it we built. And it sounds like this. So what's happening here is that the colors of the lights are determined actually by the similarity of the EEGs across these individuals, and the content of the sound is actually being created dynamically based on the EEGs of the individuals. And so it gives them some sort of a feedback that they are in a shared state. And our color convention here is blue means more similar. And so you could imagine yourself in this environment trying to make yourself blue by becoming more like your partner. Okay, so I want to say a little bit about sparsity and pattern recognition. In order to get by in life and to avoid being eaten, for example, or to find food, or to do any of the other behaviors that we need in order to survive, we have to go ahead and make sense of the world. We have to be able to anticipate things in there. And our brains, if they're evolved for anything at all, are evolved for the process of detecting patterns. I want to talk with you a little bit about what we do in order to detect patterns and how we see things. This is an extremely lovely picture. I don't need to tell you that because you see the same thing. It was drawn by Picasso in 1952. What's this a picture of, anybody? Well, it might be his wife, okay, yeah. A woman, well, he had a lot of girlfriends. What's she doing? How does she feel? What? Relaxed, yeah, okay. How does the painter feel about her?
He adores her.
He adores her, okay. There are 29 lines in this picture. I counted them. And we are able to glean all of that information almost unambiguously. Everybody in the room has the same kind of sense about this from this extremely sparse representation. So ask the question, how do we pull this off? Why does it work that way? Well, if you look inside the eye of vertebrate organisms, you will find that we have a bunch of different photodetectors, photoreceptors, which effectively respond to different locations in the external visual world. And they're interconnected in this interesting way that if light comes in, say, to this detector, it produces an output which goes out into our brain, but it also suppresses the output of its neighbor. Right? Just a little lateral suppression for this. And if you look at these stacked gray bars, I think that you will see that any interface between these two, right at the edge, it looks brighter here and darker here. Brighter here, darker here. Does everybody see that? Okay. Well, it's not, of course. I mean, these are just gray bars that are stacked on top of each other. But that enhancement of the edges is a direct effect of this simple lateral connection in the neurons in the eye. Okay? Our eyes go ahead and take shapes and turn them into lines. Okay? And it's one of the ways that we're able to resolve huge changes in illumination is because our eyes dissect the world into lines. Hence, something like that line drawing for Picasso contains most of what we would care to see. Now, in fact, our brains process all kinds of different features of the visual world, but what they do is extract relatively small discrete features on this, and different parts of the brain handle different parts of this. I meant to show this slide in the other order. This is a so-called kinesia figure. The kinesia figures have this feature that most of you will see a triangle in here, right? Even though there is not particularly a triangle at all on this screen. And this phenomenon is extremely robust, but it was recently shown that if you record single neurons in the brain, in this case of a macaque monkey, and you have so it turns out that the visual parts of our brain is arranged topologically such that a light in this part of the world shows up in a particular part of my brain back here. A light over here shows up in some other part and so on. So it's like played out as a map. If you record from neurons that would be, so if I get my light on this, I hope you guys can see this because I really can't. Okay, so here is a square on top of some circles, and here is a kinesia figure of the same. And if you record from a neuron that would be responsive to this region over here, you will see that neuron responds the same whether or not the line is there or not. Right? Our brains at this very, very early level of the brain, of processing, are actually inserting features that are not there in the image. Again, this is a process by which we create patterns in the world. It turns out that our perception and the responses of neurons in the brain seem to be largely isomorphic. And here's a kind of a fun demonstration of this. I want you to watch this figure and just stare at the center. Okay, everybody got that? Just stare at the center for a moment. Okay, now I'm going to stop it. What do you see? I mean, most people will see it expanding, right? Okay. And so that's an interesting illusion. But we did some experiments a while ago, and using functional MRI, something that Philip mentioned earlier for this. And if that perception persists for several seconds. If you go ahead and take they keep a log of how much of that perception of expansion is still taking place on the subjects after the thing is turned off, and then look at the signal intensity in a particular part of their brain, which we call in humans V5. We find that the signal intensity in the magnetic resonance images, which is a marker for cell activity, is sustained for the entire duration of this illusory perception. So this particular part of the brain is the same as our perception. And this whole issue of perception is something I'm going to want to talk about in my sort of closing slides. Paz talked about bar graphs. And I just wanted to say a couple words about bar graphs. Bar graphs were an invention. They were an invention relatively recently by a fellow named William Playfair. The one thing I want to say about bar graphs is that they are a very clever invention for us because we go ahead and interpret these lines as evidence of differences in magnitude of something in the world. Okay? And that's a really interesting thing because what Playfair has actually done is taken advantage of pre-existing models in our brain for what big and small and greater magnitude is. And these are evolutionarily built features in our brain, but this particular form of drawings, these graphs, actually take advantage of this pre-existing predisposition for how we interpret things. Philip mentioned this magazine cover, basically this is the first pictures of functional magnetic resonance imaging. This is obviously the back of somebody's head, and these colorized regions show parts of the brain whose magnetic resonance signal increased reliably when this individual was looking at a visual stimulus, right? It's kind of a very cool thing. And I love this picture, except that I made a huge mistake. And the huge mistake turns out to be the choice of yellow and red color scales. Here is another picture of functional and magnetic resonance images of the brain. Just for orientation, this is the back of the head. This is where our visual system is located. Okay? And here, right sort of around where our ears are, this is where our auditory system is located. We did two tasks with these subjects. In one task, the subject was, one task we'll say, subject was both simultaneously looking at an image on the screen and listening to things, but they changed their attention. I'm going to attend to the auditory, I'm going to attend to the visual. Okay? So given that, this picture, was the person attending to visual or auditory? Visual. Why? Red. Okay, here's the interesting thing. These are actually pictures that were taken at the same time. I just reversed the color scale. And we have a predisposition to attach high importance to red, right? Another thing that biology has given to us. And that has made a mess of my literature, because in my literature, people don't typically draw the blue. They don't typically show parts of the brain which decrease in signal intensity, right? Another thing. A few words about sparsity and pattern recognition. We know that we can compress images, and we compress things using things like JPEG or MPEG compression, which are all spectral-based compressions based on the Fourier transform of the image. I don't have time to talk about how that works, but what I can tell you is that you can compress pictures easily tenfold with no apparent loss of image quality on here. And so this is a difference image magnified by 20 of what's different between these two pictures, and you see small color shifts maybe and a little bit of speckling, but otherwise they look like they're the same picture. So if you only need, I should back up and say my camera is collecting 6 million locations on here, each of which has 3 color values, so it's acquiring 18 million bytes of data. If I can compress things without loss, why bother taking all of those pixels in the first place, right? Why did we bother making this high-density camera? Does anybody know the answer? What's your gut feeling?
Identify very small differences.
Well, can you? Between those two pictures on the top? But these are the pictures I look at, right? I mean, certainly if I magnified it, I could find some problems. Most people will tell me, nobody jumped out at this and said, because I don't know which pixels to collect, right? I mean, that seems like a reasonable thing. But in fact, it turns out that you don't need to collect all the pixels. And this is work that originated from basically this gentleman here, Stan Osher at UCLA, who showed that if you properly sample a small number of pixels from an image, you can reconstruct the image with very high fidelity. Okay? Now, what is proper sampling? It turns out that random sampling is the proper way to do this. You collect a random distribution of those things and you fill them in. And the reason this works, and this is the field that's now known as compressive sensing, is because we know some things about that picture in the first place. We know, for example, that it's compressible. Okay? Because we can compress all of these pictures. So if it's compressible, that means there is a sparse representation of it someplace. And if you actually acquire the data in its sparse representation, then you can reconstruct the image without all the original data. Here's an example of how good this gets. So this is a picture which has been heavily degraded. This is the reconstruction based on that sparsified data, and you can see that it looks like a pretty good reconstruction of the original image. Well, why do I say this? Well, what is it that we know about this picture that allows us to get to here or to here? Well, we know that it's a picture of something, right? It's of something which has some relevance to us as humans. And that is what allows us to do this trick. Our brains are incredibly good at pattern recognition. Living things have an internal life. Living things do not just have an external life. And our name for that in humans is consciousness. And there's a lot of argument about the structure of consciousness and why consciousness matters. But I want to say something that I think is really, to me, fascinating with this. Consciousness actually has material effects on the world. It changes the world. And the classical example is the so-called dual slit experiment, in which what happens is that you shine light through two slits, and you look at the pattern of the light on the other side. Now, if light were going out as a particle, what would happen is you would see two bands of where those particles collide, right? Just imagine this thing shooting grains of sand. That's what's going to happen. However, if light were a wave, what you would see is interference patterns of the light beams on one another. Now, this interesting thing happens. So this is the demonstration of what the interference patterns look like. This interesting thing happens, though, which is that when you actually do the experiment, this is what you see. Okay? You see waves. On the other hand, if you have built a device which can detect the momentum of those particles, then you have resolved something about the Heisenberg indeterminacy principle. You cannot know the momentum and location of, both the momentum and location of something. Observing its momentum alters its location. Okay? And so what happens is that if you go ahead and produce a momentum detector, it changes the output of the experiment, just detecting the momentum. But here's another weird effect. That is, if you wait to look at the picture after you've detected the momentum, it changes the result. It changes the result. And so the diffraction pattern remained wave-like until the recordings were observed. This suggests that there's some interaction of our consciousness with the world. Okay? And this machine, this momentum detector, didn't have whatever it is that thing took. Okay? A non-living system may or may not have whatever it is that makes this thing happen. I want to say just a couple of closing words about mimicry in life. And we live increasingly in a world where we are surrounded by objects that look lifelike. My phone looks lifelike. Hey Siri, what time is it in California? Oh, I have some information about that I'd like to tell you, right? Your devices have all of these crazy hallmark features of consciousness to the point that their lifeness somehow becomes ambiguous. And somebody might look at a picture like this and say, this is a wonderful, wonderful utopian vision of the future that we, just to put a heavy spin on it, that we live in a home that loves us. Okay? And that's an attractive thing. But to me, it's also incredibly chilling. To me, this is a world that has dehumanized, that has taken whatever it is about being alive away from us. And so as we move to these sentient objects, I think we always want to be considering what the boundary is in terms of what it's doing to us as human beings and as living organisms. So I just want to close with what real friendship looks like, which is different, and to thank you all for attending.